Dear viewers, greetings. In this video, let's see about the bacteria Francisella tularensis, the causative agent of tularemia, the pneumonic form of which is often lethal without treatment. General characteristic: Francisella tularensis is a gram-negative cocobacilli or pleomorphic rod. It is non-spore-forming, non-motile, fastidious bacteria. It is strictly aerobic in nature. The capsule is present in Francisella tularensis. Pathogenicity of Francisella tularensis. Disease transmission. The disease is transmitted through direct contact with infected animals. Example, inoculation of mucous membrane via contaminated ants or infected material. It may also be transmitted through ingestion of contaminated water or meat. It is transmitted through airborne via contaminated materials, include dust, hair, grass, lab specimen, etc. Insect vectors like ticks, mosquito, horseflies, fleas, lice, etc. can also transmit the disease. Virulence factor of Francisella tularensis: lipopolysaccharide capsule, FPI, the Francisella pathogenicity island, type 1 secretion system, type 2 secretion system, and type 6 secretion system. Pathogenesis of Francisella tularensis. Francisella tularensis enter the body. The capsule of Francisella tularensis appears to be the necessary component for the expression of full virulence, allowing the organism to avoid immediate destruction by polymorphonuclear neutrophil. Francisella tularensis is an intracellular parasite that can survive in the cells of reticuloendothelial system, where it resides after a bacteremic phase. Granulomatous lesion may develop in various organs. The type 6 secretion system attaches to the outer membrane of the cell and is anchored to the cell with a base plate attached to the membrane complex. The type 6 secretion system then ejects effector molecule into the target cell, which allows the organism to enter the cell, escape the phagosome, and replicate in the cell. Clinical disease caused by Francisella tularensis: ulceroglandular tularemia, the most common variety of the disease. Symptoms include ulcer on the skin that usually caused by bite from infected animal or insect, lymph glands that are painful and swollen. Oculoglandular tularemia. Oculoglandular tularemia affect the eyes. Symptoms include pain, swelling or discharge in the eye, redness in the eye, light sensitivity. Oropharyngeal tularemia. Oropharyngeal tularemia affect the mouth, throat, and digestive system. It is the form of the disease that's most often caused by eating undercooked meat from wild animal or drinking water that's contaminated. Pneumonic tularemia. Pneumonic tularemia can cause symptoms that are often associated with pneumonia, including trouble breathing, pain in the chest, dry cough. Typhoidal tularemia. Typhoidal tularemia is rare but very serious form of disease. Symptoms include enlarged spleen or liver, diarrhea and vomiting, severe fatigue, and elevated fever. Lab diagnosis of Francisella tularensis: microscopic examination. In gram staining, the Francisella tularensis appears as a tiny pleomorphic polystaining gram-negative cocobacillus. In motility test by hanging drop method, the Francisella tularensis are non-motile due to the absence of flagella. In negative staining, the unstained capsule against the dark background. Colony morphology on culture medium. In sheep blood agar, poor growth at 24 hours. At 48 to 72 hours, the colonies are 1 to 2 mm, grey-white and non-hemolytic in nature. In cystine enriched chocolate agar, Martin Lewis agar or Thayer Martin agar, colonies are 1 to 3 mm, grey-white at 48 to 72 hours. Cystine heart agar with blood, colonies are 2 to 4 mm, smooth, entire, greenish-white and buttrous with opalescent sheet at 48 to 72 hours. In McConkey agar, no growth, and in thioglycolate broth, slow with denser band near top, diffusing with age. In buffer charcoal yeast extract agar, the colonies are 1 to 3 mm, grey-white at 48 to 72 hours. Biochemical test of Francisella tularensis: catalase positive, oxidase negative, urease negative, indole negative, TSI, no gas production, H2S production in cystine supplemented medium, nitrate reduction negative, NH2 produced in liquid media negative, gelatin hydrolysis negative, Fermentation of glucose positive, glycerol positive, fermentation of lactose negative, and fermentation of sucrose negative, fermentation of maltose positive. Enzymatic reaction, beta-lactamase test positive, citrulline uridase test positive. Treatment. Antibiotics used to treat tularemia include streptomycin, gentamicin, doxycycline, and ciprofloxacin. Streptomycin is the drug of choice for this treatment. In addition, tetracycline are acceptable alternatives. Increasing research supports the use of fluoroquinolones in the treatment of tularemia. Levofloxacin and ciprofloxacin have been used clinically with success. Prevention. Not using bare hands to skin or dress wild animal. Avoid sick or dead animals. Wear clothing that covers exposed skin. Use insect repellents. Removing tricks promptly. Drinking clean water. Thank you.